Today, our speaker is Dr. Matthew Williams, Associate Professor with Department of Biomedical Sciences at West Virginia School of Osteopathic Medicine, which is one of our PUI institutions. He's also our DRPP grant awardee and has been doing research work with the support of INBRE. So, Matthew, if you want to go to your full screen and get started, the floor is yours.
Okay. Are you seeing the full screen presentation currently, or
Your slides right now.
okay, let me see if I can yeah, get the full screen going.
Yeah, you should be able to. Do you have dual monitors? If not, you should be able to hit the small box at the bottom.
Yeah, it's giving me the
Yeah, I think you're good now.
all right. Yeah, I always get confused with the dual monitors, so
Yes, yeah.
thank you so much. Yeah, thank you for the invite to be able to present my research here during the seminar. So what we work on in our lab is biotherapeutic innovations from healing springs and medicinal plants. As was mentioned, I was fortunate to receive funding to be able to pursue this project. Um, this originally started with some conversations with our osteopathic students. So sometimes I get asked, well, how are medical students considered undergrads? And isn't INBRE a lot for undergraduate education? And in the U.S., medical students are considered undergraduate medical education. So our students that we have are considered undergraduate students, at least in their professional career progression. So again, as I mentioned, this project really started with some of our conversations with students. So students were talking to me about some of the other remedies. Part of the osteopathic mindset is to think about how the body can heal itself, to think about natural healing processes. While there's definitely a place for modern pharmaceuticals, and we are definitely big proponents of that, and we like to think about our whole patient. So, not only like what is the patient doing in the clinic, but what are they doing at home too? So that questions like, well, where are they recreating? Where are they getting their water from? What kind of food are they eating? So something that's very common all across Appalachia is to recreate or even consume water from springs. So almost any day or weekend, if you drive up through the mountains of West Virginia, you can find a pipe on the side of the road that somebody's getting their drinking water from. Again, you can find people bathing in water that has a spring water source. And again, there's all these remedies that people have learned about from different family members. So again, some of those conversations started um, and started some of this project. So to get into it, my major interest is infectious disease. So I'm a microbiologist. So my goal is to try to prevent or cure infectious diseases. Again, infectious disease are still one of the leading causes of illness and death worldwide. Um, so I have seen some quotes that say, like, when we look at humanity as a whole, the only thing that has out competed infectious disease for killing humans is time itself. So especially some of the major diseases like um, malaria or tuberculosis, again, there are huge numbers of humans that have died to these diseases. Fortunately, the numbers are much lower in areas that receive modern medicine, but unfortunately, there are still locations where things like malaria and tuberculosis are a day-to-day concern. But again, infectious disease, even though we have advances in sanitation, we have vaccines against many of these infectious diseases. We've got antimicrobial therapies such as antibiotics and antivirals. There's still millions and millions of people that are affected by these infections every day, and unfortunately, there are people that die from these infections, numerous people every year. Again, this is not something, unfortunately, that we're necessarily getting ahead of, even though we've come up with new remedies for many of these infections. We have things like antimicrobial resistance occurring, so antibiotic resistance against some of our things we used to have a treatment for. Some of the bacterial infections, for example, things like vancomycin resistant bacteria or methicillin resistant bacteria. Again, there are versions of viruses that are changing, and some of our traditional antivirals are no longer functioning. But again, things like antimicrobial resistance, the spread of zoonotic pathogens, climate change, increasing population mobility, all of these things are complicating these efforts. Again, we've made great strides as humanity in reducing some of these infections. So I love this graph here because, when I first thought of reducing infections and infectious disease, my mindset was, thinking of like what had happened from like the 40s, when we started using antibiotics. The first use of penicillin as an antibiotic was really in the 40s. So I really thought of that as our major weapon that we were using against these infectious diseases. But if you notice on this graph, it's showing the rate of deaths per 100,000 people. If you notice that graph had already dropped off substantially, even by the time we got to 1940. So one of our biggest decreases in infectious disease was actually due to sanitation. So the other side of my research program that I won't be talking about today is a lot of public health and clean water access. But I do like to keep that in mind. Like, again, antibiotics are some of these antimicrobials that we're very familiar with. Again, they are great weapons. They are great therapies. But again, we did really well even prior to them with keeping humans alive. The traditional remedies that kept humans alive for millennia um, contributed something. And again, clean water access was one of the big things. But again, if we think of most modern medicine, when we think of fighting infectious disease, it's really just from like that 1940 to the 2020s that we a lot of us focus in on. But again, it's not the only answer. And again, as we see some issues with those arising, again, looking for novel therapies or different therapies is just something we need to keep in mind. Again, we've evolved in how we fight these infections through time. Again, while antibiotics, antivirals, all those antimicrobials are very, very important, we need to be thinking about what else we can do. So that's where some of this research comes in. As I mentioned, it's conversations with students, it's being from Appalachia myself, raised up here in West Virginia, drinking spring water from a pipe in the side of the mountain. My grandparents attributing it, saying, that there's some medicinal value to that. Growing up being told that, hey, we're going to drink the spring water because it's much better than the tap water. Um, there are people that kind of really attribute a lot of health benefits to this. Again, and it's fun because this project has gotten us out and to the communities and talking with individuals. And I've heard story after story after story. Just about 4 weeks ago, we were uh, grabbing water samples from one of these pipes on the side of the mountain, and this lady had a very elaborate setup in how she was collecting her spring water. And she was telling us that she had these huge jugs of water and an electric pump that she had set up with a battery so she could pump just huge amounts of water into her truck bed into those tanks. And she was telling us that she actually bathes in that water. She actually is getting enough water such that she can bathe. And she was telling us that it was her grandparents that actually brought her to that spring when she was a child many decades ago. And she even takes the water with her when she travels because she believes so firmly that that water is therapeutic to her. Again, these healing springs and medicinal plants, again, they've been used for millennia, I say centuries here, but really millennia. I mean, these are the things that kept humanity alive prior to some of our modern medicines, our modern treatments for infectious diseases. Um, Now, me being a microbiologist, I start thinking about, well, what is it about the water or what is it about the plant that could be contributing this? A lot of people, again, traditionally think of maybe the chemicals in the water. Um, so there's lots of uh, usually older documents attributing different chemicals to these the spring water especially. So that's what this excerpt here is. This is for White Sulphur Springs. This is giving the excerpt about what's in the water. So this actually broke it down. What exact chemicals are in the water. But I did notice something that from microbiology standpoint was kind of interesting. They mentioned that this, is could be, you know, it's much more potent if it's fresh. And I got to thinking, well, why would it need to be fresh? If it's just chemicals in the water, and a lot of the chemicals they were talking about, again, a lot of that should be able to be packaged. It should be able to be sent. But they always mention in these older texts about how this water can't be packaged. When people were trying to package and send the spring water for its therapeutic effects, they would say people needed to come and get it fresh. They need to imbibe the waters fresh. I got thinking, well, what would that be? And of course, my thinking is some of the microbes in the water. So a lot of this water is full of a whole microbiome. Um, of microbes, and again, some of those microbes might be beneficial. Same with the plants. Again, we attribute a lot of uh, plants as having therapeutics. We've discovered a lot of different therapies based in plants, so a lot of very valuable chemicals, uh, chemical compounds, natural compounds from plants. But and we but we kind of forget about, we realize like these plants also have microbes, but we sometimes forget about, well, those microbes themselves could be therapeutic. So again, we are all starting to get this understanding of how important our microbiome is. We all understand plants and water have a microbiome, but it seems to be like there's this gap in knowledge of like, well, how is that microbiome of the plant or of the water potentially beneficial to us as humans? So again, it sent me down a whole reading list of a lot of traditional readings. So I go over to our local library and any kind of antique store where I can find anything about herbal medicine or, um, uh, especially the spring water. So fortunately, I'm here in southern West Virginia. We're surrounded by all these different springs. We've got white sulfur springs, sweet springs, red sulfur springs, blue sulfur springs, all these healing springs in the area. And fortunately, some of these texts are at our local libraries, or again, if I start asking around or talking to people, they give me their grandmother's book that they found in the grandmother's library to look at some of this. Likewise, again, with the herbal side of it, so the plant side, again, traditionally, botany was taught with medicine. Again, there was a very close association with understanding of medical botany uh, and human medicine. And that's something that we've kind of gone away from, is that botany tie-in with medicine. So again, as I mentioned, I am fortunate to be right in the center of a lot of these really awesome old healing springs. Again, there used to be that a lot of these springs had actual hospital facilities at the spring source, and people would travel to these. So for hundreds of years, there's the history of, from the founding of the United States up through the early 1900s, of people that would travel to these springs to get drink the healing water, to take in the local plants of those that were on the grounds of those facilities. Many of these facilities would have multiple different types of spring water. So they would have like iron springs, that they call them shallow bay springs. It sounds nicer than saying they have iron water. So they'd have like the shallow bay springs, they would have the sulfur springs. Um, but again, they would also culture and cultivate a lot of plants on site at these healing facilities as well. Um, Most of them had multiple doctors on staff. They would also get scientists of the time to come in and figure out what chemicals were in the water, and they would put publications out on this. So there's this whole body of knowledge that we almost like forgot about 100 years ago. As I was reading through this again, I noticed a lot of the things that they mention in what they say that they can help prevent or uh, remedy. It's a lot of infections. So I'm thinking again with this mindset of a microbiologist of, okay, well, how are they preventing these infections, or how are these curing these infections, and what is it that can't be packaged and sent out? And so I'm thinking again about that microbiome of the water, the microbiome of the plant. So again, fortunate to be right here in the kind of center of many of these springs. So we were fortunate to go out and actually sample the waters of many of these springs. And I'll touch on some of what we found as I go through the conversation today. But again, this is not novel. I'm not the only person thinking this or realizing this. Again, we know that this source of water that you drink or that you bathe in can influence the microbiome. So there's numerous studies They're looking at this, realizing things like mud therapy. There is actually something to that. The mud therapy, again, if you're taking the microbes that are in that soil and that mud and that water, it can alter a host microbiome, can alter the immune response as well. Again, when we travel, just traveling from one location to another, that's can be enough to alter our gut microbiome. Um, Swimming in ocean water, there's benefits in going to the beach beyond just the sun. Like sometimes, we are actually altering our skin microbiome by bathing in the water at the beach or whatever your favorite recreational area is. Again, we're realizing more and more about how, the water source um, can change our disease outcomes as well. So there was a study published uh, recently talking about how, even diseases such as Parkinson's, some of these neurological diseases and disorders can actually have different outcomes depending on what the water source is. So again, there is gaining traction about this insight of water source and what's in the water can definitely have big changes in the microbiome. But again, beyond just the microbiome, also even more complex disease outcomes. So again, my main focus is to explore these natural sources for biotherapeutic discovery. Um, so what we're looking for are these live biotherapeutic products. It's kind of a fancier version of a probiotic. So a lot of people are familiar with probiotics. I'm going to talk on the difference between LBPs and probiotics here in just a moment. But ultimately, what LBPs are these live living microorganisms that we want to supplement in to a patient. It can be through the GI tract, like a lot of traditional probiotics, or maybe be even like a topical application. Um, if we're trying to prevent a respiratory infection, maybe we're actually going to get a patient to inhale these. It's not necessarily just a pill that you take or a yogurt that you drink. This, like a lot of the traditional probiotics are. Again, we're trying to have a guided, directed approach to supplementing with these living organisms. So again, one of the big questions I often get is, well, aren't you just doing probiotics research? Yes, but also no. There has been a distinction made in the field between probiotics and LPPs. So a lot of the supplements industry has really taken on that term probiotic. Um, so probiotic, again, most people have a general understanding of what we mean by a probiotic. And that's some sort of organism that has a general support of health. So it's not necessarily going to be directed at a specific infection, at preventing a specific infection or curing a specific infection. It's more about just having a maintenance of normal microbiota. Most of these are actually consumed as some sort of food product or pill formed through the GI tract. Um, as such, and many of these are actually generally regarded as safe organisms. So some common ones would be some lactobacilli, maybe some bifidobacterium. There's a lot of bacillus out there. If you ever go to the probiotics aisle and you turn around a bottle, um, you'll notice a lot of bacillus. You turn around the next bottle from a different brand, you'll notice more bacillus, but a different strain of bacillus. Same thing. A lot of these companies are just trying to market many of the same organisms, but they're saying that their specific strain is much better than their competitor strain. Again, a lot of these are regulated simply as foods or dietary supplements. Um, again, the clinical evidence and the manufacturing, again, is following a lot of food grade practices. And again, that mechanism is mostly going to be supporting uh, microbiome balance, especially of the GI tract. They state that some of that may help modulate the immune system or boost metabolic activity of the host. There are a lot of studies showing that this is beneficial. There are a lot of beneficial aspects to a traditional probiotic. Now, again, the live biotherapeutic product, these are typically going to be administered at a much higher dose. Some of these are going to be much more potent organisms, so ones that we have to be very targeted with. So it might be ones sometimes that are even listed as potentially an opportunistic pathogen if they are put in the wrong place. So just like I tell my students, like when they hear that opportunistic pathogen, it's like if you go get a probiotic from Kroger or Walmart or wherever, you bust that capsule open, you pour it in your eye, you're going to get an eye infection. same with these live biotherapy products. We're going to make sure we administer these correctly. So again, some of them are going to be much more potent. Uh, uh, Potentially they are, again, some of them that are a bit feistier. Again, we need them to be feisty to fight against our pathogens, our disease-causing organisms. So again, they might be a little feisty in and of themselves. So again, as such, they are regulated by the FDA as a biological drug. They typically are going to be going through an IND and a BLA process. Um, Again, since they are much more therapeutic, are much more potent, a little more feisty than some of these probiotic strains. Again, they actually we can show that there may be some therapeutic claims, especially if we go through the IND and BLA process and the clinical preclinical studies. Again, the manufacturing goes levels up to pharmaceutical grade for some of these. Again, because we're going to be much more potent than what you're getting at Kroger from the pill form of these some of the probiotics. Um, Again, that term probiotics, again, it's been used by the kind of the supplements agency again for a while. A lot of the supplements weren't even giving live organisms, even though they were saying they were live organisms for a while. It would come out that many of them actually weren't living organisms. Again, with a LBP is definitely a living organism, definitely a very potent active organism is the goal here. So again, it's similar to a probiotic, but typically much more potent, much more targeted, and sometimes with an organism that we have to be a bit more careful with because it is so much more potent. So overall project aims, again, a big part of my project was to train students in biomedical research. So a lot of this, my entire research lab is student-led. A lot of this was conversations with students that started it. I'm just giving them an opportunity to explore what they want to explore. Again, I've got some great medical students with great conversations, great ideas. Um, so what we set out to do is to build a library of these biotherapeutic products from our medicinal plants and spring water sources. We wanted to screen our library against the ability to antagonize or to fight against different pathogens. So we selected 5 different pathogens. So Escherichia coli that can cause some GI distress, Staph aureus that can cause a lot of different infections. So you might have heard of it as MRSA. We don't work with the MRSA version. We don't work with the methicillin resistant Staph aureus, but we work with a general Staph aureus. Again, that bacteria can cause skin infections, eye infections, GI infections. It can cause almost any infection, just depending on where it gets into the host. We also work with Serratia marcescens, which is just kind of an odd opportunistic pathogen that's a little biologically different than our other panel, so we can get a little different insights. We work with Klebsiella pneumonia that causes respiratory infections, and then also work with Streptococcus mutans. So Streptococcus mutans is a strep strain that can cause cavities, but it can also cause endocarditis. There's a lot of evidence that also shows that it can lead to poor outcomes in Alzheimer's as well. So again, we have this panel of pathogens that we're screening all of our biotherapeutic products against, all our library against. And then we started trying to investigate the ones that we found were interesting. We're starting to investigate how those actually work. So with all this, I definitely was not an expert to start off. So I've had the fortunate opportunity to travel some with this grant. I've made it all the way down to the water school at Florida Gulf Coast University. So I learned a lot down at the water school. That was at a Florida American Society for Microbiology conference last year. Made it to the American Society for Microbiology Microbe Conference in LA of June of last year. Took students with me to each of these. Did a few conferences that were a little bit different. So I did the West Virginia Mountain Medicine Weekend, where I got to meet a lot of herbalists that are actually utilizing these medicinal plants in their own remedies that they create. And then again, numerous conversations. I talk to anybody about this that'll listen, try to gain insights from neighbors, friends, family. Um, Again, almost anybody that will listen. Again, I've run into a lot of people as I'm out collecting plants or as I'm out collecting spring water sources, and it's all been very positive. Everybody that hears about this is wanting to know more about it. They want to tell me their history of how long they've used these things, what their grandparents taught them. So I've been trying to record as much of that as possible as well. Again, it's given a great opportunity to get away from the lab and play outside. We've had a lot of students get very excited about this project. we've gone to go to some beautiful places. Uh, Things like Blue Sulphur Springs. You see the student there. The fishing has the test tube beside her. She's actually fishing a sample down into Blue Sulphur Springs to collect a sample out of that. Then the Sweet Springs in Monroe County, Sweet Shallow Bay Springs. Again, just getting out into some beautiful areas. But not all this field work. We obviously bring this back to the lab, get to teach the students how to work in a biomedical lab, work with we work as a BSL2 facility, so biosafety level two. So these medical students are actually getting to see how things are developed. Again, instead of just learning about it in the textbook or in the lecture hall, they're getting insight into like, instead of it being, well, why don't we just develop these therapies against all these diseases, or how was this antibiotic created, or why don't we just prescribe this antibiotic? Again, they're getting to see the insight behind the scenes of how, all the steps that are involved in developing new therapies against some of these diseases. So progress, what kind of results do we have thus far? So um, to be able to give insight into the progress, I want to give a little insight into our process. So again, our first step, is going out and collecting these samples. That's something that's been ongoing through the 2 years of the project. Um, we start off any type of process with, again, that field collection. Um, we did have to figure out how to optimize getting our microbes off of plants. Water samples are really easy. You just collect the water sample and grow culture what comes out of the water. But the plants, the microbes like to stick to the plant. So we had to figure out what detergent would actually wash the microbes off the plant surface without killing the microbe. So we did optimize that. We had to optimize our media a bit to be able to get the maximum number of microbes off. Again, I do realize we are not getting everything off of the plants, so we're not getting everything out of the water. We are a culture-based protocol. That's something we hope to address in the future. Be able to see like more about what everything that's on these plants or everything that's in the water with some genomic studies. But currently, we've been culture based. Again, our goal is to get these bacteria that we can work with, these biotherapeutics that we can actually work with. So we've not been. We want things that are fairly straightforward to work with. We don't want things that are too fastidious or finicky. So again, we don't try to get too exotic with our media. We want to work with detergents that are straightforward. We want to work with media that's straightforward. We want to work with these biotherapeutics that are fairly straightforward. And then we do lots of screening. So screening, and more screening. This is where the students are extremely valuable. Um, and they have a lot of fun with it because they get to see fairly quickly the fruits of their labor. So we do a plate-based antagonism assay. So that's what you see on these petri dishes on the left side. So essentially, as the graphic is showing, we take a petri dish, we draw a line down the middle, and then we put two bacteria side by side on the petri dish. That second image there is showing the pathogen and an isolate as an example. So on the left side, we have some of our pathogens. On the right side, we have multiple spots of our isolate. And you see there a nice zone of inhibition with some of our pathogens. So notice that spot kind of has that clear zone, that crescent moon shape to it. Again, there's that clear zone where that pathogen has stopped growing because that isolate is somehow inhibiting or antagonizing the growth of that pathogen. Third image, same thing. That's just showing one that I'm really excited about. So that is one of our isolates. So again, that's a bacteria isolated from one of our environmental samples. And against this is against the pathogen. In this case, is Staph aureus. So Staph aureus again is really good at being a bad bug. It's really important human pathogen, and we found several good isolates that will inhibit Staph aureus. Again, we're really excited about that because Staph aureus is one that's getting a lot of antimicrobial resistance. So again, there's things like multi-drug resistant Staph aureus or MRSA, methicillin resistant Staph aureus. Again, a lot of the drugs we used to use, a lot of the antibiotics we used to use to treat a Staph aureus infection are no longer effective. But again, we've actually found some of these natural isolates that are preventing that infection, preventing the growth of that infection or of that bacteria. So then the next question is like, well, what is it we have? What is this isolate? So we do 16S PCR and sequencing to determine what those are. So one of my students made this nice image and asked that I use their image. So this is just showing what I just kind of talked through in a graphic form if you prefer that. So again, we get our environmental samples. We get the bacteria from those samples. Again, with the plants, we have to use some detergent and shake them or sonicate or homogenize them really well to get the bacteria off the surface of the plant. We do, we have looked at both like the roots and the stem portions of the plant. We obviously get more from the roots. We're not really surprised by that because we are also getting that whole rhizome. Again, we're getting some of the soil that's associated with those roots as well. Again, there's a lot of studies showing that microbiome of the roots is really potent for a lot of plants. So we've noticed that as well. We get a lot more bacteria off of the roots than the stems and leaves of the plant. Um, but ultimately, we dilute out and isolate our bacteria, take those individual bacteria, label them as isolates, um, and um, then we screen those isolates against our pathogens. When we find ones that are interesting, we send them for sequencing. Yeah, students do get insight into all this. One of the things I'm really excited about is I've got a lot of students that are realizing, hey, that's easy. It's fairly straightforward to do this PCR and sequencing. So like I tell them, like in their future, they can never have a mystery infection. So they could potentially incorporate this into their clinical careers to be able to do PCR and sequencing so that they never have a patient that has a mystery infection. So instead of waiting for days to a week to get back results on what is this infection that my patient is, they can do personalized medicine fairly quickly. And maybe they're not doing it themselves, but they can set it up in their clinic or in their hospital to be able to quickly do this PCR and sequencing in a course of a day, know exactly what pathogen is causing their infection in their patient and treat it in a personalized way. Again, this could help limit the spread of antibiotic resistance. Again, a lot of times we're using broad spectrum antibiotics to treat a infection in a clinical setting. Again, them having this knowledge that they could quickly PCR and sequence it could lead to more targeted treatment, helping reduce the spread of antibiotic resistance. So some have gotten really excited about that. Again, some of the sources of the samples we've brought in. Again, we've been fortunate to get into a lot of these local uh, healing springs. Um, I was going to put a lot of photos of some of the awesome places we've been, but then the PowerPoint didn't love it. But we've got great photos from some of these. Again, some of these are abandoned or semi-abandoned. Just amazing to see the students are just enamored by the fact that this is what healthcare used to look like, were these big resorts centered around the springs in some cases, and we've just kind of forgotten about this. The plants, we've had some botanists help us with finding this, but mostly more local herbalists have helped us with some of these. Again, we've sampled at this point over 60 different medicinal plants. One thing we realized, the reason I did quotes, air quotes there, is realized almost anything we find has some medicinal association with it. I don't know if it's just the Appalachia is just so full of them, or there's just been some sort of remedy attributed to everything around. But again, we set out to find medicinal plants. One of the herbalists that I learned from said, "You don't find medicinal plants; the medicinal plants find you." So we kind of took that to heart. The other thing I learned is you're supposed to ask the plant permission before you collect the plant. The students thought I was a little odd when I conveyed that to them, but I swear our productivity went up immensely once we started asking the plants. The students were not believe me when I said it, and then they were like, "Wait, that plant just told me no, that I can't collect it." I said, "Well, if it said no, you can't, you don't have permission to collect it." But I swear our productivity went up after we started asking the plants for permission. Some of them said no, some of them said yes. Do with that what you will, but it was just a fun little insight with the students. And again, it did increase our productivity. So again, just a few more pictures of what this actually looks like for the students that are screening through this. They learn lots of micropipetting skills. They are working in a biosafety two hood. They're learning how to do aseptic technique. They're really sharpening their eye skills. Some of them say they imagine this is what a radiologist feels like, where it's like you're looking at it, and then it's like I don't see anything, but then you look at it again, and then you get a sharper eye for seeing what you're looking for. So, for example, on the top left image here, uh, that uh, top image, the bacteria on the left is one of our isolates. The one on the right is our Escherichia coli, our pathogen. You can notice there is that little crescent moon where you're getting that zone of inhibition. Notice those two bacteria don't touch each other, so that is a form of slight antagonism right there, um, where that our isolate is actually inhibiting the pathogen. Um, so again, they say like at first they didn't even notice that, but they feel like it's looking like looking at an X-ray or a ultrasound. Whereas once you know what you're looking for, it's much easier to see. So um, on the right side is one that's maybe a bit more pronounced, a little more exciting to see something like on the right side image. So if you look at that third set of spots down, we have our isolate KG15 against our strep mutans. Notice that strep mutans, it has a very substantial zone of inhibition. It's actually polka dotted as well. When strep mutans gets stressed, it starts making these little microcolonies to where it won't fully form a nice, robust bacterial colony. So you almost get that, again, that microcolony formation there. So again, that's one that's maybe a bit easier to see what we're looking for. All total, we were able to get in the last 2 years, we have isolated 296 different bacteria from our plant and water samples. Of our 296, again, we've had pretty robust luck with finding ones that exhibit antagonism against at least one of our five pathogens. So 252 of our isolates shows antagonism against at least one. Some of them might show two. Many of them actually will even show antagonism against all five of our pathogens in our screen. To date, we've sequenced 225 of our isolates, and of those 225 that we've sequenced, we have 150 that we like to say is our candidates that we want to continue moving forward. So what makes us go from 225 to 150? Um, the other 75 there are ones that when we look at them, they might be, considered a opportunistic pathogen and maybe one we don't really want to work with, uh, at least not in the first round. We've got 150 that don't look like that they are traditional human pathogens. They're ones that maybe are just plant organisms that never have shown any infection in humans, or maybe there are actually some that, again, have been developed as probiotics in other avenues. So again, we have 150 strong candidates. Our goal originally was to look for 50. So I like to, it's nice when you can surpass a goal by threefold. So again, we have 150 candidates when we set out to find 50. Um, a lot of the strains that we've sequenced, or a lot of the bacteria we've sequenced, are definitely new strains. Like I said before, when you go and look at probiotics at the market, everybody has a lot of the same species. They're just saying their strain is better. So you might have like Bacillus subtilis, you know, KT2, and then another brand is Bacillus subtilis IGN 53. And they're just saying, well, our strain is better than your strain, and they're marketing it as such. Well, I think that we've got even better strains than they do for some of these. So we found several Bacillus strains that, again, are on the market, but I think ours are probably much more potent than what's even on the market. So we definitely have new strains that we can market as a probiotic tomorrow if we wanted to. But I think we're also sitting on some that are much more potent and could be a live biotherapeutic product as we uh, originally set out to find. Likewise, we actually have found some isolates that we're pretty sure are new species entirely. So when we do our sequencing and blast it against the database, we're getting very low percentage on our identity as when we compare it to what's in the database. So I'm pretty sure we do have some new species. Would love to be able to follow that up and see what these new species are that we've discovered. Again, what's really fun is some of those new species are some of the ones that are showing really good antagonism against the pathogens. Um, Likewise, we have been working through viability assays. So obviously, if we make these biotherapeutic products, we're working with these bacteria, we want them to stay alive. We want to make sure they are viable to actually get to the market. We don't want to have something we have to keep in the freezer or make fresh each time. So we've just been leaving some of these at room temperature. We actually have some that we just set out at room temperature for over 2 years at this point. We go see if it's still viable, and it is still viable even after 2 years. That's especially true of a lot of our bacillus. If you're a microbiologist, you realize a lot of bacillus species make spores, so we might actually just have some spore forming bacteria, but again, they are still viable. They are still, you put them into a culture, you potentially put them into a host, they're going to reinvigorate themselves and still be viable. So again, we're still working through that with many of our strains. We have a whole incubator full of them, just seeing how long we can abuse them and still use them. Just some samples of our results. We have 150. Well, we have 225 of these in our database that where we have these fully characterized. We have an isolate name, the most likely bacteria of what it is, the most likely hit from the database. We do keep track of, what our coverage value, what our identity value is when we compare it to what's in the database. Because some of these end up fairly low. So some of these, our identities are only in the 80s, or even lower. We've got some that are only 60% identical to what's in the database. So again, some of those we're pretty sure are probably some new species that hasn't been fully characterized yet. We do kind of keep track of, what some of the characteristics are of the most similar species. So for example, Bacillus cereus. Bacillus cereus is one is, it is a common soil bacteria, commonly found in food as well. It can cause some intestinal illness, but actually some strains of Bacillus cereus are what you would find in probiotics at the store. So that's what I try to give insight to the students on. Like sometimes they're like, oh, I hit on a pathogen that says it can cause disease. Well, it's like, well, it causes disease if it's misused. Again, that same Bacillus cereus that could cause disease in one situation, a different strain of it is actually beneficial. Another example would be Escherichia coli. So we all need E. coli. E. coli is hanging out in our gut, making us all nice and happy. Again, if you got your appendix still, you got lots of E. coli hanging out in your appendix, making vitamin B12 for you. If you ever have appendicitis and it goes, burst, it's going to cause an infection, and you can get severe infection from that same E. coli that was beneficial. Likewise, if you get the wrong strain of E. coli, if you're eating some lettuce that has the wrong strain of E. coli, you're going to have a bad day, and then you're going to be part of the lettuce recalls. Again, it's very strain dependent. So again, we want to investigate that more about what exact strains we have, what benefits our strains have, because I know that we have some very potent ones. We're hoping that they're very beneficial as well. So again, some of our favorite candidates, as I mentioned, we've got a lot of bacillus, just like what the probiotics market is really big on right now. But we've got some species that they're not targeting in on, and we definitely got some strains that they're not targeting in on. And again, some of ours are very potent. So those examples there that I'm showing on the right that I've shown before, some of those are those bacillus species. They're very potent against some of our key pathogens. Some other ones though that we have that other people don't seem to be playing with are our brevibacillus and our priestia and our penibacillus. Likewise, as we do initial looks at our big data set, or again, we've got 296 of these and over 200 sequenced. The majority, probably one of the most common ones are the Panthea species, especially from our plant samples. So there's not much that I've found about this Panthea species, especially in regards to human health. There's a bit of research in using it in biocontrol or microbiome engineering in plants, but nobody's made that connection that potentially this could also do it in humans. So we're pretty excited about our Panthea as well. It's thought to produce antimicrobial metabolites, so there might be some natural peptides that people haven't hit on yet that we could extract out and use as an antimicrobial. So current and future directions, we're finalizing our database. We're getting our last sequence results. I actually just got the notice right before I started this that our sequence results are in. So we're going to finalize those sequence results on like our last 20 isolates. We're going to quantify and evaluate our candidates, see what our priorities are. We're continuing viability assays on all of 150 of these that show some promise. We have started some safety assays on our most promising candidates. So, one of the things we want to be able to do is if we're proposing that we're going to put these into a human host or supplement a human host with these, we want to make sure that we can control them in some fashion, just in case they overwhelm the host microbiome. So we do want to have some insight on, like, if we were to supplement this and it gets out of control, or the wrong place, the wrong time, could we control it with our common antibiotics? So we are doing some antibiotic susceptibility testing just to see what that would look like. Then importantly, we're also working on mechanistic assays. So as I mentioned, some of these there's suggestions that some of these might produce some sort of antimicrobial compounds. Another way that microbes interact with each other. So maybe they're not secreting antimicrobial compounds. Maybe they're just that, that competitive exclusion. So we're trying to figure some of that out. Again, this is overview of how some of these live biotherapeutic products could be functioning. So one of the ways they can function is just competitive exclusion. So just having that beneficial microbe can outcompete that pathogen. So that's one of the ones I'm most excited about is just, if we establish a good microbiome with some of our biotherapeutic products, maybe prevent pathogen altogether. So a nice preventative medicine approach. Or, maybe even if somebody gets an established infection, maybe our live biotherapeutic product can come in and competitively exclude that pathogen. Another thing a lot of people get excited about is the production of those antimicrobial compounds. Again, like I mentioned, the Pentia may do that. A lot of the Bacillus species are known to do that. They produce these antimicrobials, things like bacteriocins, organic acids, hydrogen peroxide. Uh, Again, they will prevent the growth of pathogens. Some of these do immune modulation. They modulate the microbiome. They can inhibit quorum sensing in biofilm producers. They can disrupt biofilms. So we're starting to explore these mechanisms. So where I would say we've gotten thus far is just really looking at the, which of these might be secretion mediated versus which are more just that competitive exclusion. Again, I get excited about the competitive exclusion because that's what a true biotherapeutic product, a true like probiotic approach is to just supplement and prevent an infection. But we have started to look to see what do we have? Do we have any that are secreting something? So we're doing these supernatant assays with all of our candidate probiotics or candidate LBPs. Um, so again, what we do is we pellet the actual bacteria down, then we take the supernatant, filter that supernatant, and then we do our antagonism assays with that. So that's going to tell us, was there something secreted outside the bacteria? Is there going to be some sort of organic acid? Is there going to be some sort of antimicrobial peptide? Um, of the 50 that we've made it through thus far, again, we've only made it through about 50 of our 150 candidates. Only 5 of them are showing that the supernatant themselves can perform that antagonism. So again, it seems like the majority of ours are functioning to that competitive exclusion, which I'm excited about because again, that's what a solid live biotherapeutic product would do is we want it to be that competitive exclusion. Now, again, I don't want to overlook and think, I'm not excluding the ones that where there is some sort of effect in the supernatant because that might be some novel antimicrobial that we haven't discovered yet. So future directions, I did submit for another INBRE Major Award. The goals of that is to look more in depth into that mechanism of action for our 150 candidates we have. Again, further development of our live biotherapeutic product candidates. And then another thing we've really gotten a lot of interest in is what are we missing? So we've got this great opportunity to go out and look at these healing springs, look at these medicinal plants all across Appalachia. We did culture-based methods, but what are we missing? Like what else is there? Yes, we can culture these things out, but what else is part of that microbiome? So if we're fortunate to get funding, I brought on another collaborator that is his expertise is profiling the microbiomes of numerous different environmental samples. So we're really excited that we could have that opportunity to explore the entire microbiome of some of these healing springs and medicinal plants. We are working as well on getting as much of this ready and out for publication as possible. To me, though, the best result is, again, it started off as a student-led idea, student-led research project. I've had over 36 students supported by this project in the last 2 years. They've presented over 30 posters amongst them, between them at local, national, and international conference, and then just the buzz on campus about research. These 36 students have been talking to their colleagues about research and getting them interested in research. And then just to show, some of the students, again, in 24, 25, this is the list of students. We had 12 that were supported directly through either work study or the ones in bold there were actually stipend supported students on the grant. We had some volunteer students as well that were amazing. And then this is the updated list. We brought on 5 more. The grant provided stipend for students, so we brought on 5 more this summer. They're finishing up this week. They're all going to present at the INBRE conference next week in Morgantown. Again, work study and volunteer too. Again, I'm having to say no a lot more than I like to now, but we've again, over 36 students on the project, over 30 posters. Um, but then the students, like they just click. It's been amazing to see them click together on the project, um, and that buzz that's generated. Like to thank INBRE program for providing support for this project. Again, it's really about the students and the buzz that's generating that they're generating. Again, the insights they're getting, with their undergraduate medical education. They now see how some of these things that they're learning in a class, like how they can apply research to their future careers. Um, Dr. Gary Rankin is my mentor on the project. He's been great to bounce ideas off of, to give some guidance on where I need to be going, next steps. Um, Dr. Joshi, the program PI, thanks for the leadership and guidance on that. Stan Heilman is the one that reached out and gave me the invite to chat here. So gracious for the opportunity. Dr. Primerano and Dr. Salisbury giving real good insights on the next steps. So a lot of that microbiome analysis we hope to do in the future. We're hoping to work collaboratively with them to get some of those insights. And then here at WVSOM, our research office and our business affairs office keep me on track with coordinating students and budgets and all of that. So with that, thank you for taking time to listen to my presentation. I'll take any questions you have.
Thank you, Matthew. That was fantastic. So let's open up the floor for questions. You can put it in the chat, you can unmute and ask questions. I think we have quite a bit of time for that.